Our article was Requirement of DDX3 Dead Box RNA Helicase for HIV-1 Rev-RRE Export Function. Human immunodeficiency virus HIV-1 is a virus that destroys white blood cells and can lead to AIDS. Its gene expression is determined post-transcriptionally through RNA export from the nucleus to the cytoplasm, and export of HIV-1 transcripts occurs through two independent routes. Fully spliced HIV and cellular mRNA are exported from the nucleus through a CTE tap-dependent pathway. This pathway was characterized through the retrovirus MPMV or type D Mason-Pfizer monkey virus. Instead of using the Rev protein, MPMV contains a CTE, constitutive transport element, a cis-active structure located in the 3' UTR of viral genomes. Without viral proteins, the CTE recruits TAP, a protein involved in cellular mRNA transport. In another distinct pathway, unspliced or partially spliced HIV transcripts are exported to the nucleus through a Rev-RRE CRM1 pathway. The Rev protein is a viral protein that helps distinguish viral transcripts from cellular RNA. In this pathway, it binds to RRE, the Rev response element that's present in all unspliced and partially spliced HIV transcripts. This binding moves HIV-1 transcripts from the nucleus to the cytoplasm. During this process, Rev interacts with CRM1, chromosome maintenance region 1, as cellular cofactor, which allows the use of the Rev-RRE CRM1 pathway. It is suggested that the dead box protein DDX3 may also play an important role in this pathway. Dead box proteins are highly conserved and highly involved in mRNA metabolism, functioning in RNA, RNA, and RNA protein rearrangement. DDX3 is induced by TAT, an HIV regulatory protein, and located in the human X chromosome. However, DDX3 function inside of cells and its role in the Rev-RRE CRM1 export pathway is unknown. The authors hypothesize that DDX3 is important to the export of unspliced and partially spliced viral RNAs from the nucleus to the cytoplasm, and aim to characterize and determine the role and function of DDX3 in exporting HIV-1 transcripts through the Rev-RRE CRM1 pathway.
Human DDX3 is rich in serines and glycines and contains all 8 motifs conserved by eukaryotic RNA helicases, which can be seen highlighted in red in Figure 1a. To determine if DDX3 is an RNA-dependent ATPase and/or helicase, 2 DDX3 point mutants were created by independently changing lysine and glutamic acid in the ATPase motif and changing serine to leucine in the helicase motif. These became DDX3 mutant 1 and DDX mutant 2, respectively. In Figure 1b, we can see that in lane 2, in the presence of RNA, DDX3 hydrolyzed ATP efficiently. However, in lane 3 and 4, both DDX3 mutant 1 and 2 were either lost or significantly reduced in ATPase activity. Next, purified DDX3 and its 2 mutants were incubated separately with duplex RNA. Figure 1c on the right, DDX3 unwound RNA duplex in an ATP-dependent manner. Both mutations of ATPase and helicase motif abolished DDX3's RNA unwinding activities seen in lanes 3 and 4. These findings established DDX3 as an energy-dependent RNA helicase. RNA helicases can act either transcriptionally or post-transcriptionally, so it was questioned if DDX3 influenced either TAT or Rev function. First, DDX3 and its ATPase defective mutant DDX3 mutant 1 were expressed in helicells with an HIV-1 LTR luciferase reporter. Neither DDX3 or its mutant affected LTR expression, suggesting a lack of involvement in viral transcription. Next, using the Rev response RRE containing PDM128 plasmid, DDX3 was checked to see if it functioned in post-transcriptional gene regulation, which can be seen in Figure 2a. When overexpressed in Heller CV1 cells, DDX3 did not affect basal expression. However, when Rev was co-expressed with DDX3, Rev-dependent PDM128 expression increased by over 5-fold. The results shown in Figure 2 implied a role for DDX3 in Rev function. Next, the PDM128 results were verified using Western analysis of its CMV-driven GAG-pol RRE vector. In Figure 2b, Rev-dependent expression of both P24 and P55 GAG was enhanced 5 to 12-fold by co-expression of DDX3.
DDX3 in the cell modulates the Rev and RRE expression, which is required for export of GAG-encoding RNAs. DDX3 is a shuttling protein that enhances the expression of GAG proteins, indicating the presence of HIV-1. CTE is used as a control because expression of GAG proteins is Rev-independent when mediated by CTE. Therefore, on Figure 3c, we can observe in green the RRE-containing transcript being expressed in the presence of Rev, while the lanes highlighted in yellow have CTE-containing transcripts as a control. Mutations of DDX3 suppress the function of DDX3, which in turn show a reduced expression of GAG proteins in the cell as seen in lanes 1 to 6. This means that Rev-dependent pathways require DDX3 to shuttle viral transcripts in order to produce GAG proteins. Next, they wanted to answer the question of what the role of DDX3 was on Rev/RRE expression. Is it coming from increased transcription and translation? They approached the question using the same RRE and CT containing transcripts, with lane 5 in pink acting as CT control lane. In figure 3D, it is observed in yellow that there is no difference between whole cell RNA with control lane 1 that contained no DDX3 and lane 2 which had DDX3 present. Both expressed GAG mRNA. By contrast, in cytoplasmic RNA highlighted in green lane 2, DDX was present, which significantly increased the expression of GAG mRNA. Thus, increased transcription and translation was not the cause of this increased expression, but DDX3 was. In HIV-1, unspliced, partially spliced, and fully spliced RNAs fall into 3 size classes. 9 kilobase is being unspliced, 4 is partially spliced, and 1.8 is fully spliced. The goal was to analyze expression of 9.4 and 1.8 kilobase viral transcripts with DDX3 and DDX3 antisense mutant. They found there was no difference in expression with DDX3 and the antisense mutant in the whole cell RNAs as seen in figure 4A in yellow, or for 1.8 kilobase transcripts as seen in blue. But there was a difference for 9 and 4 kilobase transcripts in which the mutant sample had a 5-fold reduction in expression as seen in pink. This indicates that the export into the cytoplasm of RRE contains 9 kilobase RNA is necessary for synthesis of GAG proteins, which is verified in the Western blot results shown on the right.
To test whether DDX3 associated with CRM1 as cargo or as an integral effector, several HA-tagged DDX3 deletions were created as shown in figure 5A, which removed the nuclear export sequence from the rest of the coding frame. CRM1 was also co-immunoprecipitated with DDX3 deletions, which would help define DDX3's CRM1 interactive domain, which is portrayed in figure 5B. The image on the left in figure 5B shows Western blotting of cell lysates using anti-HA. On the right, co-immunoprecipitation using anti-CRM1 is done, followed by Western blot using anti-HA. Through this, it was concluded that the NES contributed minimally to DDX3 CRM1 binding. The major CRM1 associative determinant was found instead in DDX3 C-terminal 260 to 517 region. As shown in figure 5E, RAN and RAN Q69L were purified using nickel bead chromatography. The findings from this were that binding of DDX3 to CRM1 did not need either RAN GTP or RAN Q69L. This is shown in lanes 5 to 7, 11 and 13, and 17 to 19. Through this, we can confirm that DDX3 CRM1 interaction is different from the AN3 CRM1 cargo receptor interaction. To test whether DDX3 plays a role in the HIV-1 export pathway, human nucleoporins were immunoprecipitated using MAV414 or isotype control, followed by Western blotting with anti-HA. Specific co-immunoprecipitation of DDX3 was observed. Next, to figure out if DDX3 localized in situ to nuclear pores, digitonin permeabilized cellular nuclei was stained with MAV414. In panel 4 with GFP DDX3, there was colonization of DDX3 as a rim with nuclear pores. To test whether it was on the inner or outer side of the nuclear membrane, in situ detergent extraction was used. Through this, it was found that the signal for SUN1 and inner nuclear membrane was unchanged, while the signal for GFP DDX3 was removed. These are shown in panel 16 and 7. Therefore, we can conclude that DDX3 shuttles from the nucleus through the pore to move to the outer membrane.
Cloned HIV-1 transcript BH10 was transfected into HeLa cells with either DDX3 wild type or DDX3 mutant 1, which suppresses DDX3. DDX3 with BH10 showed the most P24 activity, while DDX3 mutant 1 showed the least production. The middle line was for baseline with just DDX3. These results indicate that ATP-dependent dead box helicase DDX3 is needed for efficient and high amounts of BH10 projection, indicating that mutated DDX3 acts as a limiter in HIV-1 expression.
It was determined that DDX3 is functionally critical for and specific to HIV-1 RNA export by the Rev-RRE CRM1 pathway. It's possible that DDX3 functions similarly to DBP5B. DBP5B is a cytoplasmic RNA helicase found in yeast involved in cellular mRNA export. It is found around the nuclear envelope and is thought to be involved in remodeling RNA cargo to allow transport through the nuclear pore complex or to facilitate the release of RNA once through. However, it functions in a pathway separate from CRM1. Also, unlike DBP5B, DDX3 alone can unwind RNA duplexes. Thus, it was suggested that Rev-CRM1 dependent export of HIV transcripts is facilitated by DDX3's unwinding of RNA duplexes. Previous studies with hepatitis C virus have shown DDX3 as primarily cytoplasmic and functioning as an RNA remodeling factor in splicing. However, with HIV-1, DDX3 was shown to be specific to Rev-RRE, not affecting splicing, but instead assisting in cytoplasmic accumulation and expression of GAG mRNA. Because DDX3 directly binds CRM1 and localizes to nuclear pores, it suggested that DDX3 helps CRM1 and its RNA leave the nucleus and enter the cytoplasm. However, it is also possible that DDX3 functions in removing cellular proteins that would prevent the export of HIV-1 RNA. DDX3 binds to CRM1 in a C-terminal region and was found to be a CRM1 specific effector rather than cargo. The difference in sequence of the C-terminal region in DDX3 and DBP5P could be due to pathway specificity. Additionally, the large number of dead box proteins in yeast could be due to specificity to separate RNA transport complexes. As such, dead box proteins may play a larger role than currently thought in remodeling RNAs in nuclear export. This article opened the possibility that small molecule inhibitors of RNA helicases could be a new group of drugs for treating HIV.
Since DPP5P works in an independent pathway from CRM1, the question of whether RNA helicase are essential actors in the CRM1 pathway has not been elucidated in this paper. However, a 2015 paper found that CRM1 and DDX3 helicase interactions occurred through RAN-GTP, in which DDX3 bound to Rev could interact more effectively with CRM1 for the HIV-1 expression pathway. Can anti-helicase molecules be used to treat HIV? This additional question is a bit controversial, as other cellular functions may be damaged, and research in vitro has not been found recently for this.
Finally, references.